তাহলে শুরু করছি সেল সিগন্যালিং অ্যান্ড সিগন্যাল ট্রান্সডাকশন। So what is cell signaling and what is signal transduction? Cell signaling means because of any molecule, when it hits a cell, it binds to a receptor. That receptor has a conformational change, and because of that change, that there is some downstream processes going, as a result of which the cell has got a response. It can divide, it can go to die, it can increase the expression of a protein, it can decrease the expression of a protein. So basically this is what you meant by signal transduction and signaling. Also signaling means how a cell communicates with another cell, how a molecule informs a cell how to give it a particular response. So it's basically communication between cells or between molecule and a cell, so that the cell shows a specific desired response. Acha. So next slide ete jaa. So cell signaling is the process by which cells communicate with each other. So it is very essential for maintaining homeostasis and responding to environmental stimuli. So if the temperature increases, a specific type of stimuli happens in our body, and that is essentially done by cell signaling. And it maintains homeostasis. So for example, if the glucose levels in your blood is high, so immediately to maintain homeostasis, uniformity, that glucose, there is a cell signaling, the glucose levels, because through insulin mediated pathway, it will simply reduce from the blood. So the key components here always are the signaling molecules, the ligand or the signaling molecules, or the drug, whatever you say, that is the main thing. The receptor, the one to which the ligand or the signaling molecule binds, and after that there is a set of pathways. Predominantly it is phosphorylation and dephosphorylation which takes place, and immediately then it goes to the level of DNA, RNA, or it goes to the level of protein, or it goes to the ubiquitin, ketone, or apoptosis, or whatever. That is the response. So three components: signaling molecule, receptor, and intracellular pathways, which leads to the cell signaling response. Next slide is out. So there are four types of cell signaling. First is autocrine. What does autocrine do? It stimulates. It releases something which binds to its own receptor, and then it does the specific response. Viral infected cell, it releases certain sort of interferons which bind to itself and the nearby cells, and it causes the specific response. Then you have paracrine, nearby cell, পাশের সেলটাকে ইনফ্লুয়েন্স করে দিল। ওকে, দেন ইউ হ্যাভ লং ডিস্টেন্স হরমোনস, endocrine system. Then you have direct cell to cell interaction. So by contact there are cell junctions, gap junctions, desmosomes, tight junctions. There are different junctions through which molecules pass. So these are the four types. So that is also known as direct cell to cell signaling, juxtacrine. So autocrine, paracrine, endocrine, juxtacrine, four types. So what are the receptors in cell signaling? Next one, you have cell surface receptors. On the membrane there are receptors known as cell surface receptors. For example, GPCRs, or G protein coupled receptors. Then you have the receptor tyrosine kinases. There are many other receptors. These are essentially water soluble receptors for water soluble molecule, or for drug molecule, or for any molecule which is not dissolving in fat. So they need to bind to the receptors for the mechanism to take place. The next is you have intracellular receptor. Inside the cytoplasm, these steroid, for example, steroid receptors. These receptors are present inside the cytoplasm. So your steroid, which for example a cortisol or any other steroids, which you have heard about steroids, cholesterol derivative. So these steroids, they pass through the plasma membrane, the fatty layer of the plasma membrane, because they are fat soluble. They go inside and they bind to these intracellular, intracytoplasmic steroid receptors. Cortisol binds to its cortisol receptors inside the cytoplasm. It forms a receptor steroid complex which directly goes into the nucleus and directly binds to the DNA and directly causes transcription, translation, whatever. So steroids work much faster. So therefore, certain drugs, but the steroids are not good for the body. Therefore, steroid drugs are to be avoided unless absolutely necessary. For example, you have a tremendous pain in your hand. So immediately they will give you a steroid. Steroid will reduce the immune system. So the pain will reduce. But regularly, if you keep on taking these steroid drugs, you will have the steroid effects in your body. That is not good. Lots of defects come because of excessive steroids. So among the cell surface receptors, you have the ligand-gated ion channels, nerve, sodium bind korlei khule jay. Ligand-gated ion channels khule jay, sodium starts to enter inside, depolarization starts to take place. Then you have receptors with intrinsic guanylyl cyclase activity. So number 2. So immediately if the ligand binds to this receptor, guanylyl cyclase gets activated and it does a downstream set of events which take place. Then you have the receptor tyrosine kinase. So the tyrosine gets phosphorylated in the receptor. Receptor is inside the cell. After there is binding to the receptor, especially in cancer, this is very important, this receptor tyrosine kinases. So the moment the ligand binds to the receptor tyrosine kinase, immediately in the C-terminal domain or inside the cytoplasm, they get phosphorylated. Then they have got SH2 domain which phosphorylated, and that phosphorylation activates a set of different enzymes of phosphorylation, and that response, either cell division takes place or stopping cell division, these responses take place. Or you have the fourth one, the G proteins, there is a coupled receptor. So G proteins are basically inside this in the inner part of the membrane of a cell, and 3 units, alpha, beta, gamma. So the alpha unit, beta, gamma unit, and the gamma, beta, gamma, and the alpha unit, they are attached to this G protein receptor. And so the moment this ligand binds to this G protein, alpha releases beta and gamma then go and phosphorylate further events. Through phosphorylation, dephosphorylation, and downstream events take place. So these are the different receptors in cell signaling. Now if you go to the next one, signaling transduction pathway. The first is you have a signaling molecule which binds to the receptor. So the first step is receptor. Then you have relay molecules which do the transduction. Transduction means taking some signal from one place to the other place. So from the cell membrane, the signaling molecule binds to the receptor. It is relayed to the cellular response. Now the signal is amplified many times. So why is it amplified 1,000 times? So one drug molecule binds to one drug receptor, but it activates several molecules of second messengers, several relay molecules. So it is amplified. Several relay molecules then again transmitted to several more. So at each step of relay it is getting amplified. So therefore the signal is amplified till you see a response. So even one molecule of a drug can cause a massive response to a drug or any signaling molecule. For example, in insulin, one problem is of course low insulin, but another problem is the insulin resistance, that the receptor number becomes reduced. So result is that even when insulin binds, it doesn't find enough receptors. So the signal is not amplified. So there is enough blood sugar doesn't go inside the cell, it is in the blood. Therefore you have high blood sugar. So it's a problem of signal amplification. Okay, then signal is integrated. All these signals which are amplified signals, they're integrated either DNA or in the protein, or it leads to the apoptosis or ubiquitination or whatever pathway, proteasome, it takes it to that particular pathway. So that is the cellular response, that is the third part. So extracellular signals are converted into cellular responses. This is signal transduction. Next slide is what are second messengers. So small molecules that relay signals within the cell. First messenger is your receptor. Okay. The second messenger is another molecule which the receptor activates. For example, cyclic AMP. For example, in the G protein coupled receptor, ligand binds to the ligand receptor, which is a G protein coupled receptor. The beta gamma unit goes and it activates the second messenger, which is adenylyl cyclase, that converts ATP into cyclic AMP, and cyclic AMP acts as a second messenger. It binds with the CAP complex, catabolite activated protein in bacteria, and then it causes this lacoperon. So even in the human being, there is a cellular response which takes place where sodium dependent glucose transport takes place, and the receptor is SGLT receptor, GLUT. So sodium and glucose, they together they go inside the cell. Rather than from the blood, it goes inside the cell. So because of the cyclic AMP, it takes place. So the moment I take a particular medicine, it acts as a ligand, it activates the receptor, activates more secondary. So even if there is one receptor, the signal is amplified several more times. So as if there are many receptors and normally it is going inside, it is mimicking. One receptor is being made to act 10 times more. So next one, these are second messenger. Now GPCR signaling, like I told you, it uses GTP, it's a G protein. So alpha, beta, gamma is attached. So then the gamma and beta unit separate, and GTP hydrolysis takes place over there. And this alpha receptor, alpha unit is bound with the GTP-GDP complex. And beta gamma goes and does the cellular response. Further phosphorylation downstream. পরেরটাতে যাও, receptor tyrosine kinases. So basically they are dimers. They are single, they are present in single monomeric forms, but when they come together they form a dimer as like a receptor. হাতটা এরকম আছে, it's not a receptor. It comes together, it forms a receptor on the top. And moment something binds to it, ligand binds to it, it autophosphorylates itself. Ligand binds, because of this specific, they two come together, enzymes are there, they autophosphorylate themselves at the tyrosine residue. Therefore, they are known as tyrosine kinase. Kinase are enzymes which phosphorylate molecules. So, they autophosphorylate. And the moment they do it, they go and do the downstream signaling, especially in cancer. এটা জাস্ট একটা এক্সাম্পল ছবি দিলাম, there are many millions of examples, মানে বলতে শুরু করলে শেষ হবে না. Next is cellular responses. What cellular responses can be seen? Gene, there is change in gene expression, there is metabolic regulation, glycolysis pathway দিকে যাচ্ছে কি উল্টো দিকে যাচ্ছে, gluconeogenesis যাচ্ছে, it depends on that. Or there could be cytoskeletal rearrangement, cell division এর দিকে যাচ্ছে, or apoptosis or survival decisions. Then different cells from the same signal, they can respond differently. The same signal molecule, it leads to a one response. In other cell, it can lead to two different types of response. In another cell, three different types of response. In another cell, it will repress the response. এটাও হয়. So feedback mechanism. So like I said, some signals, when a ligand binds to a receptor, positive response, and some will repress a response. ঠিক আছে? So say for example, in insulin, one response, one type of receptor, it will increase glucose uptake. When there is glucose is falling, then there will be another, that falling glucose will give another signal that will stop this glucose uptake. ইনসুলিন আর গ্লুকাগনের পুরো উল্টো এফেক্ট হয়। And clinical relevance, diabetes, dysregulation of signal pathway, RTKs, targeting specific drug development, specific signaling molecules, drug development, receptor গুলোকে clone করো, overexpress them in a mammalian cell lines and grow them, and then in assay plates add different concentration of different types of drugs and see what is the response. Do the assay. You can do this, drug development, it works. So conclusion, this is the most important chapter. In cell biology, say signal transduction is the most important chapter. মানে everything depends on cell signaling. If there is no cell signaling, you are dead. Okay? ঠিক আছে, আজ ক্লাস খতম, সেল সিগন্যালিংটা হয়ে গেল। পরের দিন দেখতে হবে পাম্পস অ্যান্ড আয়রন চ্যানেলসটা করাতে হবে। প্রোটিনটা বোধহয় করাবে না শাশ্বতী ম্যাডাম?